very clearly written in the textbook. Okay, so this is all I'm going to say about the regulation of the trip operon. Are there any questions?
Dr. Benny, what?
Yeah, who is speaking,
This is
please?
Alexis again. I was just curious
Yes.
about the ribosome. So is it that the ribosome falls off if it reaches the hairpin, or
No, the ribosome,
is it
no.
just the RNA?
Which hairpin? What panel are you talking about here? A, B, A, C, or D?
Just in general, like what happens to the ribosome once it reaches the hairpin again?
Which hairpin? I'm not clear
Well,
because
like in figure.
there is a stop codon. The stop codon is the signal for the ribosome to fall off. It's not a hairpin.
Oh, okay.
It's not a hairpin.
You.
You have to know that from translation. Stop codon signals for the ribosome to fall apart. The ribosome will separate into 30 and 50 subunits. It's not the hairpin. The hairpin is regulating what is happening to RNA polymerase. Okay? So you need to know that what stop codons do. Okay, any other questions? Okay, so let me then. Okay, does somebody had another question because I need to move to. Okay. So now I'm going to go into modes of genetic exchange in bacteria. That is how bacteria exchange information. In higher organisms, there is mating, and then you have all the process of meiosis where they exchange information. Bacteria don't do meiosis, but bacteria do exchange information. And it was, by Lederberg studied some of this, was the discoverer of conjugation, which is one of the methods by which bacteria exchange information. And there was a beautiful set of experiments to demonstrate, because people said, oh no, bacteria don't do any of this. So anyway, there are three modes of genetic exchange in bacteria. One is conjugation, another is transduction, and then one is transformation. In transformation, I'm talking about how it occurs in nature, not in the lab. Because when I ask you, is in lab, people think about cloning. No, cloning was not invented out there in nature. Cloning is how humans manipulate the DNAs to do manipulation of the genome. So conjugation, it is a unidirectional transfer of DNA from one bacterium to another, and you need to know these definitions. So it requires physical contact between two bacteria. That is critical. They have to be physically in contact. Okay, and this is mediated by plasmids, of which I'm going to be talking about in a moment. So here is, in a nutshell, we are going to look into more detail. Physical contact. First, there is a pilus. It's a structure that it is controlled by the plasmid, that is put out by the cell, and it grabs another cell. So when it makes contact with the recipient cell, this pilus starts shrinking, and the cells really have to be in physical contact, as shown here, where the membrane between the two cells in a very small area is going to dissolve, and these cells are going to be like one. And then the plasmid, by a process that I'm going to be explaining in a moment, is going to be moved to the recipient cell. So it goes from a donor to a recipient. The recipient does not have this plasmid, so it's going to transfer the plasmid. At the end, then this recipient becomes a new donor. This is the process of conjugation, and we'll study this in more detail. Transduction is the transfer of DNA from one bacterium to another mediated by phages or bacterial viruses. Okay? Phages are bacteriophages, bacterial viruses. So these involve phages, viruses. And in a nutshell, this is shown here. You have a chromosome, bacteria with a chromosome, a phage that has its own genome. It inserts the genome. The phage doesn't go into the cell, it's only the DNA. And then it replicates this DNA, and it will break down, not degrade. This is incorrect. This just show all this stuff that you get sometimes online. That's what happens. This is why I shouldn't do this. It's not degraded. It has to be chopped into pieces. If you degrade the chromosome, there is no transduction. You cut the chromosome in small pieces, and then the phages, what they do is they're going to package the DNA. The majority of DNA that is packaged is their own DNA, the purple. But by mistake, they can package some of this red chromosomal DNA. And then these phages then are capable of attaching to a cell. Okay, so now a different cell has a blue chromosome, just to show this. They inject this DNA that came from this bacterial chromosome. This is not a phage DNA anymore. And then this DNA is incorporated by recombination into the genome of the recipient cell. So this is in a nutshell this process. Not all phages are capable of doing this. One condition is that they do not degrade the chromosome. They cut it in small pieces, but not degrade it. Degradation means complete destruction of the chromosome. And transformation, as it occurs in nature, is the uptake of naked DNA by bacteria. Okay, this is an active process that requires proteins that reside in the membrane of the bacterial cell. And of course, you can do it in the lab, but the bacteria have to be modified to do this. So in nature, there are bacteria. Not all bacteria are capable of doing this in nature. They have to be naturally competent. And then when you do this in the lab, you make them competent by treatments with chemicals. Okay, so in a nutshell, here it is. Bacteria in nature are going to lyse. Their chromosome gets cut into pieces. Again, it's chopping up into smaller pieces. And they are going to be, here is a bacterial cell that is alive. So this cell is dying, releases DNA, happens all the time in nature. And a competent cell, which has proteins, is able to uptake this DNA, and this DNA by recombination can get incorporated here. If the DNA comes from a very distantly related bacteria, the bacterium has a mechanism of getting rid of this DNA. Okay? And that's what many restriction enzymes do. Okay, this is another view of that. I mean, I think I like this diagram a little better. So the chromosome, the piece of DNA, and then it integrates. Sometimes can exist as a plasmid if it has the conditions to become a plasmid. But most of the time it's either you integrate into the chromosome or it gets degraded. Okay, so plasmids. What are plasmids? They are autonomously replicating circular double-stranded DNA molecules. You need to know that. And some bacteria, these are very few bacteria, contain linear plasmids. The rule is this, the top one, circular double-stranded DNA molecules. There are no RNA plasmids, only DNA. And they can be present in all types of bacteria, whether gram-negative, gram-positive. And you can have a cell, here is the chromosome. A cell can have different types of plasmids, although they're shown here in the same color, but they are different types of plasmids can coexist in the cell.
Professor.
Okay, and the plasmids can be of different sizes. Some of them are small, some of them are large, medium size, etc. Yes, who has a question? Please identify yourself.
This is Johan. I was wondering why there are no RNA plasmids. Why only DNA?
Give me a